Excellent. So we're going to switch gears a little. You can go to the next slide. Can you hear me okay? Yes, thank you. So I don't have nice pictures of everyone, but our organizational committee is a core group of maybe 6 to 10 of us who have been meeting now for more than a year to try and talk about how to improve annotation and annotation tools for anyone who is interested. And our greater community is about 65 members, and we meet monthly and we talk about different annotation tools and how to progress and how to help people. Everyone.
Swish.
I think someone needs to be muted. I'm going to keep going. So anyone who is listening to this, you are welcome to join us. And the schedule is in the ERGA calendar. That's fine. You can keep going, Louisa. And our monthly meetings typically have a scientific project, and we're coordinating to try and be part of the BGE project, the Community Annotation Task, which is Work Package 9. And I'll mention at the end, but we're going to the Biohackathon in October. Okay, next slide. So I thought I would use these few minutes to talk about a tool that we have been putting together with the idea of demystifying annotation for anyone who wanted to start doing this on their own. And so this is a screenshot of a part of the PDF we have created. You can get it on the web page or with this QR code. It's very easy to find if you go to the Annotation Committee on the ERGA webpage. So I'm just going to go through a couple of steps, not the whole thing, because it's there for you to find. And the first step is to listen to everything Nadej said. So you can go to the next click, Louisa. And before you start, make sure you're finished assembling. There are tools out there, in addition to what Nadej talked about, to check for completeness and accuracy. Make sure your haplotypes are collapsed. Make sure you've removed contamination and organelles. We also strongly recommend that you make your assembly and your data public. And doing this will also make it visible to put it into pipelines that exist in these databases like Ensemble Rapid, or to request annotation at NCBI. So make sure you're ready to start annotation is our first step, and this is in the flow chart. Next slide. And then we recommend for anyone who wants to do their own annotation that you then go out and gather the available evidence. You can click, Louisa. And this is well detailed in the outline as well. And people have been emailing and asking us for the amount of all of these different types of data. And this is a sort of thing we would like to discuss with you. But right now, I will just talk about the types. You want to have RNA-seq or some sort of expression data from your species. It does not have to be the same individual. It should be as many diverse tissues and developmental stages and ages as possible. But any one of these is also better than nothing. And in small animals, we often just put the whole thing in, right? You also need to gather protein evidence for this annotation, but this does not mean you need to do protein sequencing. So a lot of people are only using BUSCO genes from their organisms, or all of a well-curated protein database from your phylum. And there are ways of finding this without doing protein sequencing. And then if you have iso-seq data, this is also useful, but a lot of people aren't generating this, but this is long read RNA-seq data. Next click. The next step in the process that I also cannot go into depth about is to predict repeats. So you can click, Louisa. And this is an essential step in any annotation, whether you're interested in repeats or not, which I think are very interesting. At the very minimum, you need to identify them and mask them in your genome before moving on. And we recommend several tools for this, but I won't go into any more detail than that, and we can keep going in our tour. And then the most important step, of course, is ab initio training and gene prediction. So you can click. And this is really where you find your genes within the genome. This also includes upstream and downstream regulatory elements and the intron-exon structure. And We recommend several different pipelines to combine for this. And it really should combine and include both an ab initio training, meaning some sort of computational prediction of the gene structure that is based on signals and known structures, but not sequence similarity. And then this should also include a gene prediction step where you're basing your genes on matches to known genes. So you have this computational ab initio part, and then you have a similarity-based mapping of gene prediction. And then these are all combined in one of many ways, depending on your pipeline. Next slide. And then the very last step is the evaluation of your assembly or your annotation. So at the very least, you should run Busco to evaluate the completeness in your predicted genes and compare this to what you find in your genome. It's also very useful to compare with closely related species and to look at the numbers of expected genes and to have an idea of what you would expect for GC content and numbers of genes, types of repeats, this sort of thing. But for many species, this is not available. And so we really do think that we need more comprehensive tools for this. And I'm putting this here if anyone would like to work with us to generate this. Next slide. And this leads right into my last and most important point of being here is that at the end of the month, we are going, a group of us, to the biohackathon in Barcelona, and some of the members of this activity are here watching, I think, where we are going to test out three different annotation pipelines using both model species and our own taxa to see if we can develop containerized and very user-friendly pipelines that we can all start to use on our taxa. So if you would like to join us and try this out, you can get in touch with me and you can join the hackathon virtually. So we're putting a call out for anyone who is interested in learning a bit more and bringing their perhaps weird animal to this to see how this performs on diverse organisms. And then at the end of this, we are working on this discussion of what priorities we can make for evaluating our annotations. Next slide. And so in the future, we plan to keep meeting monthly like we have been, to make these pipelines available as they become complete with the, hopefully after the biohackathon. And we are also have a group joining us at the biohackathon to feed these into Galaxy, which I think will make it nicely available for a lot of other people. So maybe you have something to contribute, and please get in touch if you do. And I think that's my last slide. Yes. So thank you again. Rob has put the link to the biohackathon into the chat. And so please get in touch, and thanks for listening.